Hi, can somebody confirm you can hear me?
Yeah, I can hear you.
Thank you. Sorry for being a bit late. I forgot once again, I have to go into Zoom and I don't remember my account information, so I had to look that up. And anyways, that's why I'm a little late. Another reason why I'm a little late is if you can take a look at my clock here, you probably can't see it now, but
Yeah, that's what.
my clock is not correct. Let me share my screen here. Before we begin, are there any questions? I do need to know, is there anyone in attendance who is trying to add the class?
I am. I would love to join if possible.
And who are you?
Madeline Clems. We've been emailing back and forth.
Okay. Yeah, you were the one I missed getting the first time.
No worries.
Just a minute. Let me stop sharing my screen, then I can see this better. Oh, how do I do that? I'm just going to write down Madeline. And what's your last name, Madeline?
It's Clems, Klemz.
Okay,
Thank you.
thank you.
Oh, I'm also trying to join the class. My name is Shiva Bali.
Yeah, Shiva, I've already got you down here.
Oh, okay. Thank
We
you.
talked to you on Tuesday, but thanks for letting me know you're here today. I'll make a note of that. Okay, where was I? I was trying to get the syllabus up. Chapter 1. It came up even though I already had Word up. All right, today is July 13th, and we're supposed to be covering Chapter 1 and Chapter 10. It'd be really nice if we finish that. I don't know if we can. There is a lab today. We will cover lab 1 later today, whatever the time the lab is, 7 o'clock, I think, in the summer. Any questions about what we're going to do? Note that you have a plagiarism quiz, which is due this Saturday. And I don't know how I'm going to do that for the people who are trying to add the class because you don't have access to Canvas yet. We'll have to deal with that. For the lab, I can email you the lab. And so if you are in attendance, just email me to send me a reminder that I need to send you the lab. Or if you're in the lab, you can just tell me after I finish lecturing in the lab. And then we have our first practice quiz next Wednesday. Any question about any of that? The practice quiz says it's out of 0, but you'll get extra credit point. I think you get 1 extra credit point if you get all the answers correct on the practice quiz. It's not much, but it's a little bit. I do encourage you to take the practice quiz because you need to have Respondus Lockdown Browser running and active on your system before you take my quizzes. And it's no sweat off my back if I delay your taking the practice quiz, okay? But if you're more than 2 days late taking quiz 1, you're not going to be allowed to take it because the answers will be showing after 2 days. So you want to make sure that Respondus Lockdown Browser is running and active on your system. And I've got a link on my Canvas website for where you can download Respondus Lockdown Browser. Every other term, there's one student who has trouble getting that to run on their computer. And if you do have trouble, you should contact the computer help desk, and they will help you change the settings to get Respondus Lockdown Browser running on your computer. You can let me know, but I won't be able to help you. All right, any questions about any of this? If not, let's go to chapter 1. Why did that come up? Yep, I don't want help. What do I want? I want 2 here. All right, so we had talked about where you can find microorganisms and answered it that it's almost everywhere on the planet Earth. Where is everywhere? Environments where microorganisms are found are on the land and in the water, and you can actually find them in the air as well, and they can be found up about a mile up into the air. And on the land, you can find microorganisms down about a mile into the soil. If it's bedrock, it may not go down a mile. Under the ocean floor, down about a mile, you find microbes on plants and animals, essentially anywhere where liquid water is present. We do need liquid water for microbes and all living life forms to engage in metabolism. But microbes can be found between a minus 20 degrees Celsius and about 120 degrees Celsius. Remember, 0 degrees Celsius is when water freezes, and 100 degrees Celsius is when water boils. So we can find living organisms beyond 0 to 100 degrees. And below 0, microbes make antifreeze, and so they can be chemically active. But below a minus 20 degrees Celsius, all microbes, even if they have antifreeze, will freeze, and they cannot engage in metabolism unless water is liquid. And so that may not kill them, but they cannot grow. They cannot be metabolically active, but they can be alive while they're frozen. And then above boiling water, we do have some microbes that do live in that temperature. And the highest, I think, was actually 120 degrees Celsius that we've ever found a microbe that could actually, I don't know if it grew in that temperature, but it could survive that temperature. And that was in the Marianas Trench, the deepest place on Earth under the ocean. And that's why it was that temperature. And that is, that's the temperature that water will boil at under the deepest part of the ocean, the Marianas Trench. And that's where we found microbes tolerating those temperatures. Any question about any of that? Microbes are numerically the most abundant life form on Earth. In 1 gram of soil, we can find about 10 to the 8th microbes. I'm talking about bacteria here, not viruses. If I were to talk about viruses, all the numbers would be increased by 10 times if we add in the viruses. But if we're talking about bacteria and other microbes like yeast, we have about 1 times 10 to the 8th microbes in a gram of soil. This will depend on how much moisture is in the soil and how much organic matter is in the soil. The more moisture and the more organic matter, you can go above this number. And then the drier the soil and the less organic matter, you can go down below this number. Like in about 1 gram of the soil in the Sahara Desert, we don't have 1 times 10 to the 8th microbes. We only have about 1 times 10 to the 6th microbes per gram. Any question about any of that? All right. In ocean water, do you think in 1 gram of ocean water, which is 1 milliliter, do you think you have more or less microbes than in 1 gram of soil? Anyone have a guess?
My guess is more.
Your guess is more, and that's a reasonable guess. But actually, the oceans are one of the deserts on the planet Earth. In the ocean, we actually find less microorganisms as well as less organisms than about any place else on the planet Earth. The deserts are one of the deserts of the planet Earth. And so actually there's less. In 1 gram of human feces, normally we have more. In 1 human being, normally have more. And then on the entire Earth, of course, we have more. In 1 gram of feces, we have about 1 times 10 to the 11th microbes. It depends on how fresh and how wet the feces are. But when a feces comes out of a person, over 50% of the weight of that feces, and that may be dry weight, I don't remember. About 50% of the weight of the feces is actually from microbes in the feces. Okay. And if you, somebody were to go to the bathroom in the desert and it would become very dry, it would be less microbes in the feces at that point. So like I said, it does depend on how fresh it is and how wet it is. Generally, when people have diarrhea, they have lots of bacteria in it. Let's take a look at the numbers. Ocean has about 1 times 10 to the 6 microbes. Like I said, the ocean is a desert compared to the soil. And actually in the desert, like the Sahara Desert, you only have about 1 times 10 to the 6 microbes in the 1 gram of soil in the desert. Human feces can range between 1 times 10 to the 6. That would be a very dry feces, like somebody goes to the bathroom in the desert and then it dries out. We'll only find 1 times 10 to the 6, which is the same as the desert soil, by the way. And like I said, very wet feces, we can find 1 times 10 to the 11th, which would be, is that number there? 1, 2, 3, 4, 5, 6, 7, 8, 9, 10, 11. 55% of the wet weight, I got it right there, is of feces is actually microbes. On 1 human, we have about 3.8 times 10 to the 13th microorganisms. In the entire Earth, we have around a nonillion. That's 1 times 10 to the 30th prokaryotic cells. Now, if we were to count viruses, it would be 1 times 10 to the 31st. Like I said, with viruses, you just multiply by 10. The human population in comparison, it's getting close to 8 billion. That's close to, a little less than that. 8 times 10 to the 9th. As opposed to on the planet Earth, how many microbes we have. Microorganisms can cause disease. If a microbe does cause disease, we call that a pathogenic microbe. Pathogen means an organism capable of causing human disease. We're talking about human diseases here, not animal diseases. I mean, we do have like cat pathogens and cow pathogens, which can be the same as a human pathogen, but they can be different. So only a small percentage of all of the microbes on the planet are actually pathogenic. Only a small number compared to all the microbes on the planet can cause disease. When we're talking about disease, it's important to distinguish between infectious diseases and inherited diseases. Some examples of an infectious disease are COVID-19, the flu, salmonella, typhoid fever, for example, Staphylococcus aureus, skin boil. Those are some examples of diseases caused by an infectious microorganism. So all infectious diseases are caused by a microorganism or a parasitic worm. Any question about any of that? All infectious diseases are caused by a pathogenic microorganism or a worm, because a worm isn't quite a microorganism. It does fall under the umbrella of a microorganism, and we'll talk about that in a little bit. So those are infectious diseases caused by microbes. Inherited diseases are one that a baby or person are born with. What is an example of an inherited disease? Anyone have an example?
Sickle cell anemia.
Yeah, that was a very good one. That's actually the one I was going to mention, because you should have learned about that one in your preliminary biology class. Sickle cell anemia is a disease a person gets if they are born with 2 alleles for sickle cell anemia. And this is an inherited disease. It is not caused by an infectious agent. Infectious diseases can spread from one patient to another, but not all infectious diseases can spread. For example, I'm losing my mind here, so just a minute. I'm trying to remember what disease I'm talking about. Botulism does not normally spread from one patient to another. You have to eat or consume Corynebacterium. No, that's diphtheria. Corynebacterium, no. I'm not remember what agent causes botulism now. Something botuli, but Mycobacterium. Is that it? That causes Mycobacterium pneumoniae, causes pneumonia. That would be an infectious agent that can spread from one person to another. But tetanus, for example, generally does not spread from one patient to another. You have to get tetanus usually by a puncture wound where the microbe gains entry into the body and then grows anaerobically, and that's why it has to be usually a puncture wound. And you get that from outside, and it does not usually spread from one patient to another. It is possible for those diseases caused by a microorganism to spread, but the only case I can think of is if you have a cannibal and he eats someone who has tetanus, the cannibal could get tetanus. Okay, but that would be the only case I can think of with tetanus spreading from one patient to another. And I suppose that's the reason why you should not go out and eat other humans. Any question about that? I must have went backwards. Yep. All right, so here we're looking at some microorganisms. You can see there are various shapes and sizes. There's the size right here. This looks close to E. coli, which is a little bigger than the average bacteria, but just a little. The width is actually about average. The length is a little bit longer than the average bacteria. Microorganisms are organisms that are small and we need a microscope to see them, meaning a microbe we cannot see with our human eyes. There's a microbe here, and this is a microbe, and actually this is these red things are viruses attacking a lymphocyte. That's a microbe. This is mold and the hyphae of one particular cell. Come on, mouse. So this would probably be one particular cell right there, and then the mold growing together. This right here, let me see if I can blow that up. This is a microbe, but this is an unusual microbe because it is colonial. And see each of these dots? That's one cell. And then these here, that's a baby colony inside of a mother colony. And this mother colony has 6 babies. This is called volvox. It'll open up and then release the babies, which will then go out and form a colony in another location. And then the mother colony can continue living. Any question about any of that? Many microbes are individual cells, almost all bacteria, all yeast, protozoa, but not all microbes are unicellular. Some, like volvox, are multicellular. Okay, microorganisms play many beneficial roles to both humans and beneficial roles on the planet Earth. Microbes decompose organic waste, and they are probably the main thing that decomposes organic waste. You can have fungi and mushrooms also decomposing organic waste, and fungi are actually very important, mold, but mushrooms are not quite as important, but a mushroom is definitely not a microorganism. Microorganisms are also important in that they're producers in the ecosystem. We have both photosynthetic microorganisms which produce carbon. They fix carbon, like carbon dioxide in the air, and they fix it and make glucose into it, which green plants do that. But we have green or photosynthetic bacteria, which do that too. And then we have chemosynthetic bacteria, which are producers in the ecosystem. And it's true that they take CO2 and make carbon, glucose, for example. Chemosynthetic bacteria we'll talk about in a little bit greater detail later, but they get their energy from a chemical compound. They do not get it from light, for example, but they are producers. And all of the organisms which engage in chemosynthesis, meaning they derive their energy from a chemical compound, are prokaryotes. There are no eukaryotes which engage in chemosynthesis. Microorganisms are involved in the nitrogen cycle. Microorganisms produce a lot of industrial chemicals, such as ethanol that you drive in your car, acetone, if you've ever used that in a chemistry or biology lab. We can use microbes to make insecticides. We use microbes to make fermented food, and that includes things like vinegar, cheese, sourdough bread, and then beer. And then any leavened bread using yeast actually is slightly fermented. It's just that the alcohol from the yeast, it's not very much. And then when we bake the dough, the alcohol leaves. And there isn't very much, so it's not very acidic, so we don't even notice the acid. We use microorganisms to make many products used in manufacturing, such as cellulase. We use microorganisms to make antibiotics, like penicillin G is made by a microorganism, and that microorganism is actually the Penicillium mold. So penicillin is made by the mold Penicillium. And then we use microorganisms to make other products used in medicine. For example, insulin. We pulled out the human insulin gene from a human and then put it into a bacteria to make the insulin. And then we grow up the bacteria, which makes the insulin. We purify out the insulin, and then we give it to the diabetic patients. Microorganisms are important in biotechnology. Most people don't quite understand that biotechnology is the use of microbes and organisms to produce foods and products for human use. Biotechnology is actually centuries old. It is not something new. Like when we ferment cabbage to make sauerkraut, that is biotechnology. The same thing when we ferment grape juice to make wine. That is biotechnology too. Genetic engineering is a new technique of biotechnology. Usually in genetic engineering, we take a gene out of one organism and put it into another organism to get a useful product. Applications of genetic engineering include the producing of a variety of proteins from bacteria and fungi. And I already mentioned this is how we make the human insulin gene. We got it out of a human who had the correct sequence for the human insulin gene, and then put it into bacteria, and then grew the bacteria, isolated out the insulin, and then it's sold as a pure product. We can use genetic engineering to engage in gene therapy. Gene therapy is when a child is born with defective genes, and we put in the correct genes. Most of the time, these are children born with a defective immune system. They're lacking a gene to have a complete immune system. And you can take out their immune system cells and grow them in vitro, and then add in the correct gene, and then inject the cells back into the patient. That does give a child a functioning immune system for about 3 or 4 months. The problem is that the cells that you've injected into the child will not last forever. Cells have a half-life in the human body, and the immune system cells do not live that long, only about 3 or 4 months. But then you can repeat the gene therapy, and that's what some children have been doing. The last I had was the very first patient, which this is probably about 10 years old. She had now aged to about 20, so she's now a young adult, and she has a functioning immune system because every 3 or 4 months they apply gene therapy, giving her the correct gene, and then inject those cells into her. And so now she's probably someplace around age 30. And then another application of genetic engineering is genetically modified bacteria, where we change the bacteria to make something we want. And I did mention that we've done that with making bacteria to secrete the human insulin gene. Any other questions about any of this? All right. let's talk a little bit about microbial ecology. Bacteria recycle nitrogen on the planet Earth. We've already talked about that, but bacteria help recycle other elements besides nitrogen. They help recycle carbon, they help recycle sulfur, and they help recycle phosphorus. It is true that each of these has a geochemical cycle where carbon moves from one part of the planet Earth or ecosystem on the planet Earth, and it's switched by a microbe or some other organism, and then it can move to another part of the niche of the microorganism. So carbon, sulfur, and phosphorus have cycles where the element is converted from one form to another. And it is true that these cycles do not have as many steps that microorganisms are involved in recycling the nutrient on the planet Earth, but nonetheless, for all of them, microorganisms are important for recycling these elements on the planet Earth. It's not like the nitrogen cycle, like where almost every step happens because of a microorganism. With these cycles, carbon, it frequently can change from one form to another, like when a rock is degraded by erosion. That's not a microorganism doing that. And then that would release the carbon into the environment. Nutrients are also recycled on the planet Earth. When an organism dies, microorganisms and yeast microorganisms and fungi, especially the mushrooms, can degrade that organism, and that will release the nutrients for other organisms to use. Any question about any of that? Anyways, carbon, nitrogen, sulfur, and phosphorus are all examples of microbial mediated biochemical cycles that help support life on Earth. And it was first discovered by Winogradsky and Bajering, who showed that the reason why nitrogen moved from one place in the ecosystem to another and changed from one form to another by microbes. This was first shown by Dr. Winogradsky, and he did his work from 1880s to 1892. You don't need to know the years almost ever. When I give you the year, I'm just giving you the year to put it in a time frame for you. Any question about any of that? All right. Microbes can also be important to human welfare because microbes can engage in bioremediation. Bioremediation means life and to remedy, such as these workers are spraying an oil slick sludge, meaning there's an oil slick and this is oil. They're spraying the oil with microorganisms to help degrade the oil and then metabolize the oil from oil to carbon dioxide and water. We did mention that microbes are important in the breaking down and recycling of nutrients in sewage. Without microorganisms, we would not be able to break down sewage. Microorganisms and bacteria are important in handling pollutants. We can use microorganisms to make the pollutant either less toxic. For example, mercury can be made less toxic when we use bacteria to convert the mercury into a less toxic form. And then we can use microbes to I don't know, clean up the pollution. Like when we have an oil spill shown here, you use microbes to digest the oil. And if all the oil is converted into CO2 and water, that's basically fine for the planet. It may not be fine for humans on the planet because you're increasing the CO2 in the atmosphere when you do that. But so far, we've never had an oil spill that went beyond, I don't know, several miles. Any question about any of that? All right. We can use microbes to make biological insecticides. Some microbes are pathogenic to insects, and they can be used as an alternative to a chemical pesticide. One reason for using them as an alternative is that chemical pesticides are toxic to humans. So when someone sprays the pesticide, you don't want to eat anything from whatever he sprayed, the tree or the vine. At least initially, you want the pesticide to break down, and then it will be safe to eat the organism or the whatever you treated with the organic pesticide. Anyways, an organic pesticide is less toxic than a man-made pesticide. We can use microbes to prevent insect damage to crops, as well as to help prevent disease transmission. An example of using a microbe to treat something is using Bacillus thuringiensis on apple trees. Bacillus thuringiensis is fatal to insects. So Bacillus thuringiensis, when it gets on a microorganism, it'll tend to grow and then kill off that organism. Any question about any of that? All right, but Bacillus thuringiensis is harmless to humans and animals. It is not toxic to us, even if we consume them. And there we're looking at Bacillus thuringiensis. My father, as early as the 1960s, treated his apple trees with Bacillus thuringiensis. And he did find that it did work. It's just that he was fairly lazy and he only treated the apple trees one time. And even when you're using a chemical pesticide, you need to use that pesticide about every 2 weeks, because the pesticide breaks down. And it's true that Bacillus thuringiensis also breaks down. Plus, my father applied the Bacillus thuringiensis to his apple trees in Western Oregon. And as you know, in Western Oregon and Western Washington, they get a lot of rain, and the rain would wash the Bacillus thuringiensis off the apples. So I can tell you, in the 1960s, when my father used Bacillus thuringiensis, we did get fewer worms, but you do need to repeat it, the treatment, every 2 weeks, as well as after every rain shower or every rainstorm. And my father was way too lazy to do that. He only did it one time. And I'm not sure why he only did it one time, probably because he never read the directions. But even with the chemical pesticide, you have to apply it every 2 weeks before it'll be 100% effective. Any question about any of that? All right, if there's no questions, let's move on to the naming and classifying microorganisms. This is a switch, but we're doing chapter 1. And in chapter 1, we are moving from topic to topic and not spending any time on many of the topics, if not all of the topics. We will come back to many of these topics and talk about them in greater detail. But in chapter 1, we're just introducing a number of the topics. This man here is the first person who established a system of scientific nomenclature, and he actually gave organisms a name. And he also was the first person to start classifying organisms into different groups. So you would say something like the elephant and the hippopotamus are similar, that they're very large mammals. I guess I should say elephant and the rhino, that's a little closer. But the point is they could be put into a similar group, and they certainly both belong to animals. I don't know if they belong to any other group. Well, they're both ungulates. They both eat vegetation. Surprisingly, this individual's naming system is still used today. His classification scheme is no longer used, but he's the first one who started classifying all organisms into different groups. Anyone know this guy's name? Nobody's going to guess?
Isn't it Linnaeus? Is that, do I have that correct?
Yes.
Okay.
Linnaeus or Linnaeus. His correct name is actually Carl or Carly. I don't know how you say that Scandinavian name, Linnaeus, but he liked Latin so much that he changed his own name to Carlus Linnaeus, and that's just a Latinized version of his name. Okay, and for some reason our textbook calls him Carl Linnaeus, meaning they Latinized his last name, but not his first name. Anyways, he named all living organisms, or started to do that, and he gave each organism two names, the genus name and the specific epithet name. The specific epithet name gives the species when you give them the genus. Like when you go sapien, that doesn't mean man. And I don't remember what it is, but sapien is used on some other genus that isn't human. And I don't remember what it was, but there's at least one animal which is called something sapien. It might be a monkey or something, but so to name a species, you give the genus name. For humans, that would be the Homo genus, and then you give the specific epithet for that species, and that would be sapiens. And so Homo sapiens together names the species. So each species name has two names. They're always Latin names, but the good news is it's used, the Latin name is used wherever you are in the world, whatever language they're speaking. So if you say Homo sapien, anyone in the world should know what that means, because that is the term that is used for humans. This system is called the scientific nomenclature because we name everything. It's also called the binomial nomenclature because every species has two names, by two normal names. So binomial means two names. Any question about any of that? All right, there are some rules when you're using the scientific nomenclature. The first, besides that everything has two names, is that the names are either italicized or underlined. So for example, whenever I am typing it on the computer, I will usually go back and italicize a scientific species name. However, if I'm writing it on the board, I can't write in italics, and so I will write it and then underline it to illustrate that it is a scientific name. Any question about that? The first letter of the genus of a species name is always capitalized. Conversely, the specific epithet name is always lowercase. So these are showing you the capital H in Homo and sapiens is lowercase, and that's italics. And if you can't do italics, you spell it out and then you underline it. All words are Latinized and they're used worldwide. For example, if you wanted to talk about Homo sapiens, you could just say Homo sapien. You don't know the name for the country you go to. You can just use Homo sapien and everyone would understand you, at least if they know scientific terms. E. coli or Escherichia coli is the same. And then Staphylococcus aureus is also the same. Each word either is descriptive or it honors a scientist. Like Homo sapien means wise man, that's sapiens, and Homo that walks on 2 feet. I don't know how wise we are, but we are, I suppose, the wisest animal. Escherichia coli is both descriptive and honoring a scientist. Coli is part of our colon, and this is one location that E. coli grows in, at least in humans. So coli is descriptive. Escherichia is named after Dr. Ascher, who was an early microbiologist. So that's honoring a scientist. Staphylococcus aureus is totally descriptive. Aureus means golden, and Staphylococcus aureus is golden in color, the cells. And then it grows as a ball or a cocci, and so we call it coccus. And then it grows, the cocci grow as a cluster of grapes, and that's where staphlo means. Staphlo literally means cluster of grapes. Any question about any of that? After first use, scientific names may be abbreviated with the first letter of the genus and a period, and then the specific epithet spelled out. So for example, Staphylococcus aureus should be spelled out Staphylococcus aureus the first time you use it in a paper, but thereafter you may abbreviate going S period aureus. And the same thing as Escherichia coli is E. coli. Let me state that E. coli is so familiar. Everybody who are biologists knows what E. coli is. And so this is one where even among scientists, they do not always spell out Escherichia the first time they use it. And I'm sure you are all familiar with E. coli. You know why that's so familiar. And so this is an exception, Escherichia coli. Usually, even among scientists, they will always call it E. coli. Any question about any of that? All right. When we are classifying organisms, we're going to talk about the classification of microorganisms. However, it is true that all living things are classified. So let's look at the classification of all living organisms on Earth. The first division of all organisms is into the domain. We have a 3-domain system. The bacteria, the archaea, shown in yellow, and then the eukarya, shown in blue. The domains are based on genetic data. So the domains are never argued about which domain belongs in which. And whenever we discover a new species, you can simply sequence the DNA from that species and then determine which domain the organism lived in. And the reason for that is because when we have our trash cans, we won't put them all together. I mean, we could if we had all trash, but I'm saying the trash is not usually put right next to the recycling or the cardboard. They're usually in separate bins. The point is that the three-domain system, also called the tree of life, because if you put on a trunk here, this branches like a tree. And then that main branch branches into other branches, and that's the way a tree branch branches. Any question about any of that? All right, so there's no argument among scientists about what a domain is because we have a domain defined. And then there's no question that the organism belongs in that domain because its sequence of DNA will be more similar with any other member in this domain than any member outside the domain. Any question about any of that? So the domains are defined and they are based on the genetic sequence of the organism. Usually, the genetic sequence is determined from people who got rid of the outer dead layers of the peptidoglycan, and then they only have a fresh tube. You put that tube in water and plug it in. Obviously, you keep the instrument out of the water. And you sequence the organism, and then you'll know which domain the organism belongs to. Any question about any of that? All right. So we have three domains, bacteria, archaea, and eukarya. You can then further divide those domains into a kingdom. We're going to talk about the six kingdom approach, where bacteria is both a domain and a kingdom. And the same with archaea, it is both a domain and a kingdom. But the prokaryotes are a domain, but they can belong to different kingdoms. Eukaryotes or domain eukarya can be separated into plants and animals. They can be separated into fungi, and then they can be separated into protista. How we separate these kingdoms is we just say a kingdom is something smaller than a domain. The trouble is scientists have never solved how many domains there are and which organism, excuse me, not how many domains there are, how many kingdoms there are and which organism, which domain the organism belongs to. Because you can answer that question just by sequencing the DNA and then determining which domain the organism belongs to. Any question about any of that? Let me check the time to see how we're doing. Come on. My mouse died. Come on. There we go. All right, we go to 6:50. Oh, we got quite a long time still to go. All right, the point about the kingdom is the kingdoms are not defined. Scientists argue about which organisms belong into which kingdom. And then scientists argue about how many kingdoms there are. As I mentioned, we're going to use the 6-kingdom approach, but there are different numbers of kingdoms. Actually, there are some people who say there's only 3 kingdoms, in which case the kingdom will be the same as the domain. There's some people who say a 5-kingdom approach, and I'm not going to talk about it. Then there's 6 kingdoms. And what you do is 6 kingdoms is you take the domain eukarya, and then you divide it into 4 kingdoms: plants, animals, fungi, and then protista. Let me briefly mention about protista. A protist is an eukaryote that it does not belong to a plant, an animal, or a fungi. So any eukaryote that's not a plant, not an animal, or not a fungi, we put it into the kingdom protista. But the point is that the protista are not organisms that have a monophyletic origin, meaning there's different organisms in the protista, and they are not, those different organisms are not necessarily closely related. Any question about any of that? For example, the plants and the euglenosa are not that similar to each other when we sequence the DNA, but they are both photosynthetic, or at least most of the plants are, and the euglena are definitely photosynthetic. But plants did not directly evolve from the euglena. They had another common ancestor, and that common ancestor is something down here. Any question about that? So the protista is a mixed bag of different organisms that are not plants, not animals, not fungi, but they are belonging to the domain eukarya. How many kingdoms there are depends on who you ask. As I mentioned, there's a 3-kingdom approach, the 5-kingdom approach, the 6-kingdom approach, which is the approach I'm using to discuss the kingdoms with you. And this is the kingdom approach we will use. And it's actually the kingdom approach that most biologists use too. But you could go larger, splitting the kingdom protista into different kingdoms. And that would be the 9-kingdom approach, the 15-kingdom approach, and I believe the 20-kingdom approach. Now, when you get up higher in numbers of how many kingdoms there are, you can begin splitting things that we wouldn't think would normally be split. For example, the plants can be split into 2 different kingdoms, depending who you ask. And they're obviously not using the 6-kingdom approach. They're using something like the 20 or more kingdom approach. You also start splitting the archaea into different kingdoms if you have like over 20 different kingdoms in the domains. And then you start splitting the bacteria into different kingdoms as well. Any question about that? All right, your textbook talks about something that they call a cell type, and bacteria have the cell type prokaryotes. The cell type is another name for superkingdom. And superkingdom is something greater than a kingdom, that's why it's called supra, but it's also greater than a domain. And why we use the term superkingdom today is because, at least since the 1960s, maybe the 1950s or even 40, somebody came up with the term superkingdom, and it is a group which is actually larger than the domain, the group of domains. And the reason being is the cell type prokaryotes is found only in the bacteria and the archaea. And then the eukaryotes have the cell type eukarya. The cell types differ. We're not going to get into that now, but the eukaryotes have a nucleus and then membrane-bound organelles. And the archaea and the bacteria, which are prokaryotes, do not have those organelles. Any question about any of that? All right, let me shrink that a little. Let's talk a little bit about some of the characteristics of the domain bacteria. All of the domain bacteria are prokaryotes. We can find peptidoglycan as the main molecule in the cell wall of bacteria. So if the bacteria produce a cell wall, it's going to have, sorry, I just lost a blank what I was trying to say. If the bacteria have a cell wall, then the main molecule of the cell wall will be peptidoglycan. The domain bacteria multiply by binary fission. That's simply where the cell may grow in size, meaning get larger in size, and then it enters cell division. And binary fission is the way most bacteria grow or reproduce. And that's when literally one cell divides into two. Before the cell divides, it has to replicate its DNA. And then the DNA molecules move to opposite ends of the cell. And then the cell literally divides in two. We have the domain bacteria, which can use all energy sources. They can get their energy from an organic chemical, such as glucose. And that should be familiar to you because that is where humans get their energy from, an organic chemical such as glucose. However, there are some bacteria which can get their energy needs from an inorganic chemical. And these would be the I'm dropping my names now. This will be the chemosynthetic bacteria which engage in chemosynthesis, getting their energy from an inorganic chemical compound, such as hydrogen sulfide gas or ammonia, substances which are toxic to us. And then there are some bacteria that can get their energy needs from photosynthesis, where the bacteria can harvest light and then take the light energy to use that energy for the cell's needs. Usually the cell will pull down CO2 and bring in water and then make glucose from CO2, water, and then the energy that is given off by the molecule degrading. Any question about any of that? Most bacteria are either free-living decomposers or primary producers. So most bacteria do not cause us any harm, and they actually help the environment by recycling nutrients, or they help the environment by fixing carbon, bringing in carbon dioxide from the air, and then making glucose. And then the glucose is available for other organisms to eat. And all living things on the planet need producers to make food. Even if you say, well, I don't eat green plants, so I'm not benefited. Yes, you are, because you're eating something like cow, and the cow is eating the grass. And the only reason why the cow has energy to give you in the meat is because the cow got the energy from eating grass. All right, most of free-living decomposers are primary producers, but some bacteria are parasitic and some are capable of causing disease, and these we call pathogenic. Some bacteria are pathogenic. It's only a small number or small percentage that are pathogenic, but there are some which are pathogenic. Any questions about the domain bacteria? If not, let's move on to the domain archaea. The organisms in this domain, there we go, are all prokaryotic organisms. Some of the archaea are found in extreme environments, and this was the first location that we actually found archaea. So we first found archaea in environments where there's lots of methane. We found them in environments that had a high, very high level of salt, like the Dead Sea between Jordan and Palestine. Some of the archaea are extreme thermophiles, but initially, your textbook author used to state that the archaea are found in extreme environments. That is not correct. The archaea are found in all environments on the planet Earth, and they make up a significant proportion of the bacteria in cell, and I shouldn't say bacteria, the prokaryotic cells in soil, as well as the significant proportion of the plankton are archaea. And none of the archaea are pathogenic. And so after this lesson, we're not going to be talking much about the archaea. And that's simply because the focus of this class is to discuss microbes that cause human disease. And there is no archaea which is known to cause human disease. Any question about any of that? If not, let's move on to the domain eukarya. There are 4 kingdoms in the domain eukarya: the plants, the animals, the fungi, and the protista. I guess I forgot to mention that with the archaea. Of the archaea and the eukarya, none of these cells or living organisms have peptidoglycan in their cell wall. Of course, animals don't even have a cell wall, so of course they don't have peptidoglycan. But only in the bacteria do we find peptidoglycan. And it does not help prevent getting sunburned. But there is no peptidoglycan in eukarya or in the archaea. The eukarya have different kingdoms. Here we're talking about the kingdom fungi. This involves the multicellular molds and the multicellular mushrooms. It also involves the molds which are not multicellular, and all of the yeasts are not in the multicellular molds. We do have some of the fungi which are unicellular. This includes all of the yeast. The yeasts are always unicellular. And the fungi are important in decomposing things on the forest floor or the ocean floor. That reminds me that the idea of dumping human waste into the desert, into the ocean, isn't a good idea, because it's not going to decompose very quickly. And then very deep, the deeper down you go, the less microbes you'll find. In general, we're talking about the ocean. That's not the case like when you go to in a pond, which I suppose the highest concentration of pond would be the surface of the pond. Not the surface, the underneath of the pond, whatever you call that, the bottom layer of the pond. All right, any questions about the kingdom fungi? Let's talk about the kingdom Protista, the protists. This kingdom has at least 3 different groups within it. There are some protists that are plant-like, and these are the algae. There are some some protists, which are animal-like, and these we call the protozoa. And then there are some fungi or protists, which are fungus-like protists, and these we call fungus-like protists. And I'm not going to talk about them any further. I can tell you one example of a fungus-like protist is the potato blight, which actually ruined the potato crop in, I think, the late 1800s. And it led to mass migration of Irish to the United States, because if they stayed behind in Ireland, then many of them would have literally starved, and there's nothing they could do about it because there wasn't enough food around. All right, the algae have cellulose in their cell walls, very similar to green plants. The algae can engage in photosynthesis. They do produce oxygen when they engage in photosynthesis, and they can make glucose. And that would be important for anyone who eats algae. The protozoa, or the animal-like protists, these include the amoeba, the paramecium, and the euglena. They absorb or ingest organic chemicals for their food. So they cannot make their own food, they must ingest their own food. And oftentimes they get that food from a cell that they kill and then digest, or it could be they're getting it from, I don't know, the environment. Somebody's left like glucose on their hand, and then the microbe on your hand will start using it. That's actually my wrist, not my hand. The protozoa may be motile. If they are motile, they can be motile by use of flagella. If you remember, in prokaryotic cells, if they are a motile prokaryote, they have to have a flagella, because that's the only thing prokaryotes have to be motile. But eukaryotes have more than just the flagella to be motile. Some eukaryotic cells are motile because of flagella, and with humans, there's actually 2 examples that I can think of. One is a sperm cell is motile because it has a flagellum, and then the trachea in the upper regions of the upper respiratory system have cilia, which constantly beat up, bringing anything from down in the respiratory system up further in the respiratory system until it's dumped in the mouth, where upon that time it'll be either spit out or I guess taken and eaten. Let me go back to the motile protozoa. They can move by flagella, but they can move by pseudopods. For example, it looks like this cell is moving this way, sorry, my mouse isn't working, moving this way. And so the cell is putting out more cytoplasm here and then pulling in cytoplasm here. And that's how the cell can move by pseudopods. Protozoa or eukaryotes can also move by cilia if they want to. A cilia would be a hair-like structure smaller than the flagellum. And generally the cilia beat in unison for giving maximal movement. Like if this volvox, which has cilia on, I think there's one cilia on each of the cells, at least on the outside. And if it wants to move that way, it'll then beat this cell, not beat, the cilia will beat this way on that one will beat this way, this one will beat that way. And the ones at the front may not beat because they won't exactly make it move this way. Any question about the kingdom Protista? Oh, come on, mouse. All right, let's talk a little bit more about the kingdom plants. Kingdom Plantae can be divided into 4 different groups: the mosses, the ferns, the conifers, and then the flowering plants. All of the kingdom plants are multicellular, and we're not going to talk any further about kingdom plants because the plants do not cause any human diseases, meaning they do not directly cause an infection because they're not pathogenic. And I suppose you could say that when a plant does make pollen and then you get a hay fever, I guess that could be considered a disease. But other than that, plants do not cause any known disease. And so we're not going to talk about them because the focus of this lesson is to talk about microorganisms that hopefully someday you will see when you get your first job. Any question about any of that? All right, if not, let's move on to the kingdom Animalia, the animals. We can divide the animals into sponges, worms, which are called scientifically helminths, insects, and vertebrae. All of the members of the kingdom Animalia are multicellular. There is no animal cell, which is only one cell and then remains one cell. Any question about any of that? Oh, let me state that when we group things into one grouping, the worms or the helminths, that is in reality a human error, meaning we think all worms are together, so we tend to lump them together. But in reality, the platyhelminths are as different from the flatworms, that's the platyhelminths, are as different from the nematodes as they are from humans. So that the genetic relationship between the flatworms, the platyhelminths, and the roundworms, the nematodes, is so different that these two should not be in the same group. It's only humans that think they look together, they should be together, and we place them in the group helminths. But in reality, all the helminths, I shouldn't say all the helminths, but all the different groupings of the helminths, meaning the platyhelminth and the nematodes, which do have pathogenic members, as well as the annelids, which is a worm, but that's the earthworm. And there's no annelid which is known that causes human disease. So the point is we're lumping them together, but in reality, these should be broken up into Probably 3 different categories. Any question about any of that? All right. So what do we mean by microbiology? And what is a microorganism? What falls under the umbrella of a microorganism? All right. I'm going to use the term microorganism to mean an organism. So when I'm talking about a microbe, which is not an organism, and specifically that would be viruses and prions. These are not organisms because they have no cells. And prions are not even alive. All they are is a protein molecule. So they're not alive. And viruses, you can argue about whether they're alive or not. We're not going to get into that argument. But they are unique in that these microbes are not organisms. So I will not use the term microorganism to mean viruses and prions. However, your textbook author and many other authors do not do that. They will use the terms microbe and microorganism synonymously, and viruses and prions are not organisms. And I suppose it's okay for me to call them microorganisms. It's just because they have the word organism and they are not an organism. I do not like using the term microorganism to describe a virus and a prion. So I will use the term microbe to describe viruses and prions. And then bacteria and archaea are both microorganisms and microbes. There are certain of the eukarya which are also microorganisms. All of the protozoa, all of the yeast, most of the molds, some of the algae are microorganisms. And then the helminths, the parasitic worms, are under the term microorganism. And you're going to say, wait a minute, most adult worms are not something that you need a microscope to see. Most worms I can see with my naked eye. And that is true. However, the helminths, which are multicellular animals, belonging to two, what's the word I'm going to? Not family, but clade or order. Whatever's under kingdom, King Peter's son. So King Peter's son. So phyla would be the next one. So these are different phyla, roundworms and flatworms. The worms, the adult worms, are not microorganisms, at least most of them. There might be 1 or 2 parasitic worms which are microbes, but all the other parasitic worms as an adult are not microorganisms. You can easily see them without the use of a microscope. For example, the Taenia saginata, the beef tapeworm, is something like 30 feet or maybe 30 meters long. Definitely not a microorganism. However, some of the life stages of the helminths are microscopic. And generally, the stage of the worm which infects a human is microscopic. So if the human is infected with fertilized eggs of a worm, that will be microscopic. And there are a few of the helminths which actually burrow through our skin, but they are microscopic. So usually the life cycle that infects a human is almost always microscopic. Any question about any of that? Oops, wrong one. All right, let's talk a little bit about viruses. We will talk later in a whole lesson about viruses, but I just want to introduce the topic. Viruses are acellular. They consist of either DNA or RNA as their core molecule, and this will be the genome of the virus. And they either have DNA or RNA for their genome. They never have both DNA and RNA. It's either one or the other. That's a little different from cells because cells always have DNA for their genome. But we've already stated viruses are different, and this is another way that they differ. The core molecule, meaning the viral genome, is surrounded by a protein coat that makes the coat of the virus. Some viruses have a lipid envelope surrounding the protein coat. And when does a virus replicate? A virus replicates when it is infected its host cell, and it only replicates inside its host cell. The host cell is a cell that the virus can infect and then replicate in. Outside of their host cell, viruses have little or no metabolic activity. Viruses are not part of the three-domain system, so we do not separate them into domains. Any question about any of that? All right, so let's take a look at the three domains of cellular organisms. Domain bacteria, domain archaea, domain eukaryotes. The cell type for bacteria and archaea are prokaryotes. The cell type for eukaryotes is eukaryotic cells. The chromosome of bacteria is generally one chromosome, and that chromosome is in a circle like this, so that the place where the chromosome starts is the place where the chromosome ends. For bacteria, there are no histone proteins on the DNA. In eukaryotes, in contrast to bacteria, we do have the presence of histone proteins on the DNA. And eukaryotes generally have more than one chromosome. Usually eukaryotes have many chromosomes, and the chromosomes are not circular, the chromosomes are linear. For example, looking at the X chromosome here, the place where the X chromosome begins is different from where the X chromosome ends. Any question about any of that? The archaea are a little bit in between for the bacteria and the eukaryotes, and we're not going to talk further about that. The ribosomes of the bacteria are smaller than the ribosomes of the eukaryotes, and here the archaea are the same as the bacteria. They both have ribosomes of 70S in size. The S stands for sedimentation coefficient, and that is where the ribosome sediments in an ultracentrifuge gradient when the ribosomes are put in an ultracentrifuge. The eukaryotes are a little larger ribosome, and it will sediment deeper down in the ultracentrifuge gradient. The cell wall between eukaryotes and bacteria differ. Mainly in bacteria have peptidoglycan. The eukaryotes do not have peptidoglycan. Any question about any of that? All right, if not, let's talk a little bit about a brief history of microbiology. When we're talking about the history of microbiology, you should understand that this largely includes the debate over whether spontaneous generation could occur and whether microbes spontaneously generated or if they came from a pre-existing cell. There's a few other topics we'll talk about in the history of microbiology, but like I said, in the history of microbiology, especially early in its history, it largely concerned the debate whether microorganisms could spontaneously generate. When we're talking about the history of microbiology, there's a term, the golden age of microbiology, covering from about 1857 to 1914. You do not need to know this date. It's just early in the age of microbiology. The golden age is when essentially whenever anyone looked at something under a microscope, they were looking at something new. So whatever you looked at, there was something to write up, and it would be something new, and people would be interested in reading about it because it would be something that nobody knew before you discovered it. Any question about any of that? All right, here's a few of the things we'll talk about. The golden age of microbiology being this period here, and that would be from the start of microbiology, which I don't even know if that's shown here. Yep, too far. Yeah, it doesn't really say when the first discovery of microbiology was, but sometime close to this point, it might have been a little bit earlier. We'll talk about, actually, the first microbes were actually discovered by Van Leeuwenhoek. First observation of live microorganisms. So I guess 18, is that right? I can't read that. Is that 1873 or 1673 would be the start of microbiology? 1673. And the golden age didn't happen until 1857. All right, let's move on, talk a little bit about the history of microbiology. When we're talking about the history of microbiology, I will be giving you a year. Let me get that out of there. But except for this first date, you do not need to know any of the dates. I'm only giving you the date to be, I don't know, a timestamp, something to talk about, but you do not need to know the date except for this very first one. You do need to know that a little over 3.5 billion years ago from today, we have fossil records in rock of the first ancestors of bacteria. And this is the first evidence of life on Earth. And that's 3.5 billion years ago. Then the next subject I want to talk about is 4,300 BC. Babylonians actually wrote written records of microorganisms making beer. And this is unique because the Babylonians wrote down recipes for making beer, but they did not know that there were microbes making the beer because they could not see the microorganisms that made the beer. And then the next written record we have is 600 AD, where the Mayans wrote down records for making a fermented beverage from cacao, which to us would have been a chocolate. Chocolate is actually fermented, and the first written record happened at 600 AD. And the Mayans, of course, could not also see the microbes, but it was the microbes that made the chocolate. You might not have known the chocolate was fermented, but it is actually is fermented. Oh, let me go back here. You should know the order of events on the slide. So before the Mayans could have written their record, you should know that there were the first written records of microorganisms by the Babylonians. And then before that, we had to have obviously life on the planet Earth. And so you should know the relative rank of these items. That the Mayans writing the records of microorganisms engaging in making chocolate, that had to happen after the Babylonians and after the first living life occurred on the planet Earth. Let me shrink that just a little bit. The first observations of microorganisms could not have happened until the invention of the microscope, and Janssen developed the compound microscope in 1590. Surprisingly, Janssen did not use it to take a look at living things or microorganisms. I'm not sure why Janssen developed the microscope, but he did in 1590, and it had to be developed before anyone could look at living organisms. Surprisingly, the first person to use the microscope and then use it on something that was alive was Robert Hooke, who in 1665, once again, you don't need to know the date, he looked at plants and he saw little boxes that he called cells. Let me open up Paint and explain where the term cell comes from. Hopefully I'll be able to draw here. In the monastery, the monks had private rooms where they did their monk things, probably slept in these private rooms too. And they called these rooms cells. And when Janssen looked at cork or shavings of cork under the microscope, it looked like this, which were the cells in the monastery. Now in the monastery, we'd have a hallway right here, and then there would be a door into each room right there. Okay, but when Robert Hooke looked at the shavings of cork, which was the bark of the cork tree, which is obviously a plant, he saw these structures that he called cells. And that's where the term comes from the term that the monks used to describe their rooms. They called this their cells. Any question about any of that? All right, how do I get rid of this? Robert Hooke looked at a few other living things, and he saw cells in all plant matter. Like I said, he first found it in the shavings of cork, but he looked at other plants and he found cells. He did not find any cells when he looked at animal tissue. And to tell you the truth, from 1665 for about another century, nobody found cells in animal tissue. And the reason being is that animal cells only have a cell membrane, which is much thinner and less easy to view under a microscope than the cell wall of a plant. And so the reason why Robert Hooke found cells in plants, but not in animals, was simply that the primitive microscopes were fuzzy and could not focus well on a cell. And so when they focused on tissue of animal cells, they could not see the cells. It was just too fuzzy, the microscopes. And it took 100 years, further development and improvement on the microscope before people were able to look at animal tissue and then see cells in the animal tissue. From Robert Hooke's observation, it marked the beginning of the cell theory, which is that the theory of the cell theory is that all living things are composed of cells. The cell theory was developed further by Virchow. It says my internet connection is unstable, so I might be coming in and out, but that's the first use of a microscope on living things. The above are not specifically microbiology, like the development of the microscope is nothing to do with microbiology. And then the cells that Robert Hooke was looking at were not microbes. They were plant cells. Any question about any of that? I think we're now done with the lecture. Let me check on the time. Yep, goes to 6:50. So I think I'll get ready to stop here because this is a good place to stop. And we didn't quite finish this lesson. How many more slides do I have? I do have a number of more slides to go. So we're a little behind schedule. Let me go with one more slide. Nope, I want to stop here. All right, any questions? Oops, that's the wrong one. I'm going to save that down. All right, if there's no questions, I'm going to end the lecture and I will see you at 7 for the lab.
Thank you, Professor. See you
All right.
then.
Bye.